Hi, today I would like to tell you about a new app that our lab developed. So the app name is L1000FWD. It stands for L1000 Fireworks Display. So this is the interface for the app. You can see the URL is at amp.farm.mssm.edu/L1000FWD, all caps. And what you see here is a map, a visualization of 17,000 L1000 gene expression signatures. And the similarity between signatures is determined by the cosine distance of the expression vector of the differentially expressed genes. The layout of the plot is achieved with the Allegro force-directed graph algorithm that is also used in Cytoscape. This is an alternative algorithm that we believe that is as good or even better than t-SNE. So in this current view of the map, the nodes are colored by their mechanisms of action, their known mechanisms of action. You can see that most of the nodes in the map are gray. That means that those are signatures for drugs that don't have known mechanisms of action. So the exciting thing about this visualization is that we can predict directly the mechanisms of actions of a small molecule just by seeing how it clustered with drugs that already have known mechanisms of action. So for example, if we zoom into this purple cluster, we can see that some of the drugs have known mechanisms of action that is represented by the purple color, and those are tubulin polymerization inhibitors. So those are typically used as chemotherapeutic drugs to prevent from the cells to proliferate or to divide by destroying their tubulin filaments that are used for mitosis. However, we also see in this cluster a lot of gray crosses, and these represent small molecules without known function, and those known functions are likely to be tubulin inhibition or some type of an inhibitor of the tubulin polymerization process. We can also color the nodes in the map using other properties. For example, we can color by clustering. So those are simply coloring by an automated algorithm called BD-scan. We also have a k-mean clustering visualization as an alternative automated clustering and identification of clusters on the map. We can color by the cell type that was used in the experiment, and you can see that some clusters are very unique to a specific cell type, while other clusters are cell agnostic. For example, this cluster over here has representation of all cell lines. So that means that those drugs, regardless of the cell line, they cause the same changes in gene expression. While in this green cluster, the cellular response is very unique to this particular cell type, which in this case is VCAP. We can also color by time point, and you can see that the distribution of time point is not random. This coloring is based on the phase of the drug, whether it's in preclinical, already approved drugs, so the orange ones are approved drugs, whether it was withdrawn from the market, or it's one of the clinical trial phases. And we can also color by chemical features such as rings, scaffolds, number of rings, number of scaffolds, and predicted mechanisms of actions. One of the unique features of the app is that for drugs that are FDA approved, the orange nodes on the map, so by clicking on one of those points, we are directed to a signature report page that provides the up and down differentially expressed genes, the chemical structure of the drug, links to external information about the drug, the predicted mechanisms of action, as well as the diagnosis and the usage of the drug, the prescription of the drug, and the prescription with other drugs collated from the Mount Sinai electronic health records. And in this plot we show the age distribution of the users of the drug. Finally, the website provides an ability to submit gene expression signatures that can be queried to identify where your own gene expression signatures fall within the space of those drug perturbations. So by submitting a query, I can see the drugs that are most similar and those drugs that reverse my up and down differentially expressed genes for my own experiments. And the detailed results can be downloaded by clicking on this blue button. So this is the list of the most similar signatures, and this is the list of the opposite signatures. So this is it for the demo of the L1000FWD.